Alright class, today we'll be looking at part 4 of IGCSE Biology paper 6. We'll be looking at enzyme activity. Let's start by looking directly at two different types of enzymes catalysts we can make use of in this experiment. The usual experiment is on dissociation of hydrogen peroxide, and two catalysts can be responsible for that: biological catalyst and non-biological catalyst. The biological catalyst usually that is commonly used is called catalyst, and this can be found naturally in fresh potato. But non-biological catalysts like manganese 4 oxide is usually supplied as a chemical. So the steps to follow will be the same for all the processes we'll be looking at. Let's take note of those steps. So we add 3 cm cube of hydrogen peroxide to a test tube. Then we add fresh potato strips and shake the tube gently. The test tube is either covered with thumb or with a stopper to prevent it from spraying off. Now, this time around we are using fresh potato. Some other times we may change the source. We may decide not to use fresh potato. We may decide to put manganese 4 oxide, as you will soon see in the subsequent slides. But what matters at the end is the enzyme catalyst is meant to break down the hydrogen peroxide to form two products, as you can see in the equation: water and oxygen. Now, the oxygen gas is a testable gas here. It's very easy to test. Of course, bubbles formed show that a gas is formed, and then you can test it by using what we call glowing splint. When a glowing splint is used to test for the presence of oxygen, it will relight to show that oxygen is truly present. So this is the positive test when we use catalysts, the ones that are naturally present in the potato strips. So let's look at other controls that we can use. Let's look at use of manganese 4 oxide instead of the catalyst naturally present in the potato chips. So we use everything, we follow the same steps, but instead of potato chips, we use manganese 4 oxide. We will still get a positive result. That's why we call it positive control. Okay, we get positive result because bubbles of oxygen will still be formed, and it shows that reaction are taking place, and it's fast. But we can have negative controls. Let's look at what happens when we use a boiled potato chips. When you use boiled potato chips, of course, a fresh potato chip is expected to have the enzyme catalyst present and active. But in the case of a boiled potato chip, or potato strips, sorry, we discover that you've expose the potato to extremely high temperature as high as 100 degrees Celsius, right? So when you do that, the enzymes have been must have been denatured. So for that reason, there is no reaction, so no gas will be formed, no oxygen will be formed, so the lighted splint will not be relight, the glowing splint will not be relight. Okay, then what happens in the control 2, negative control 2, when there is no hydrogen peroxide? Everything else is there, no hydrogen peroxide. Instead of hydrogen peroxide, we replace it with water. Again, there will not be any reaction because there is no substrate for the enzymes to work on. So yes, there is fresh potato, yes, there is catalyst, but there is no hydrogen peroxide. So catalyst cannot act on water, cannot break down water. Okay, so there is no reaction. So let's look at the third control where we have a cold environment. Enzymes work best at a particular temperature we refer to as the optimum temperature. But when the temperature is extremely low, the enzymes are less functional. Okay, so that is the reason why, yes, there are gases formed, but very little, so the reaction is very slow. So these are the different patterns, the different ways this experiment may proceed. If a question comes testing our knowledge on all of this, it will now be very easy for us to deal with them adequately. So thank you so much. As we round up the class, remember, please help us to like, share and subscribe to our channel so you can get notified anytime we have another video for you. And we appreciate you as we round up this class and prepare for the next. So join us for the next video, factors affecting photosynthesis. See you. Have a nice day.